Alright, so in this video we're going to be talking about photosynthesis. And photosynthesis can be described by the equation 6CO2 plus 6H2O, which yields glucose and 6 oxygen. Alright, so looking at this diagram here, essentially the two main parts of photosynthesis are going to be our light reactions and our dark reactions, also known as carbon assimilation reactions or Calvin cycle. Alright, so if we look at the equation here, that we can see that H2O is a reactant along with carbon dioxide and ultimately produces glucose and oxygen. So in this diagram, we see that in the light reactions, the electrons from water are taken by photosystem 2 and produces oxygen as a byproduct. And then in our dark reactions, the carbon dioxide enters and ultimately it is assimilated into a carbohydrate known as glucose. Alright, so for the light reactions, the electrons from water are kind of passed down an electron transport chain similar to that of in the mitochondria with cellular respiration. And those electrons are passed to NADPH. And NADPH, just like NADH, is an electron carrier. And similarly to cellular respiration, when those electrons in the electron transport chain go through, it creates a proton gradient. And the proton gradient occurs, or the proton gradient will have an impact in generating our energy, ATP, that's needed for the Calvin cycle or the dark reactions. It's important to note that the dark reactions don't mean that it occurs at night. It can occur during sunlight as well, but it does not need the light to occur, as with the light reactions need the photons from light. But also it's important to note that without the light reactions, the carbon assimilation reaction of the Calvin cycle will not proceed for a very, very long time. So although photons of light is not required in the Calvin cycle, the energy that's produced in the light reactions is needed for the Calvin cycle. And so without the light reactions, the carbon assimilation reactions will not go on for a long period of time. All right, so next we'll talk about where all this occurs. So the main organelle is a chloroplast that this occurs in, and the light reactions occur in the thylakoid membrane, which is the membrane that's in the thylakoid in this diagram right over here. And so that's inside the chloroplast. So this whole thing right here is chloroplast, and then inside we have things that are known as thylakoids. And so light reactions occur in the thylakoid membrane, and then the dark reactions just occur in the stroma. So we'll take a closer look at each of the two main processes in photosynthesis. So we'll start off with the light reactions. And light reactions, the main goal of the light reaction is to basically produce NADPH, which is an electron carrier, and ATP. And like I said before, these two are very important in the Calvin cycle. Okay, so the process by which this occurs in the thylakoid membrane is what is known as a Z-scheme. So this diagram here shows kind of how the electrons transfer through all these electron transferring complexes and proteins, which starting off at photosystem 2, which is P680, exciting the electron, going down to P700, and then exciting the electron again, and then eventually ending up at NADP+, which is which gains the electron and becomes NADPH. So that's the whole process. And we can see here that in this diagram to the right is kind of a more of like a cycle of what happens is, so the photons from the light will excite this P680, and once the P680 is excited, the electrons will leave, and another electron has to come in to get it back to P680. And so the question is, where does this electron come from? And to answer that question, it comes from water, which is right here. So P680, when it loses that electron, it becomes a really good oxidizing agent, and the electrons it takes are from water, and eventually the byproduct of that will produce molecular oxygen. So that's the overall Z scheme. So starting off at photosystem 2 and photosystem 1 here, these electrons will travel through the Z scheme, go through these plastoquinone, cytochrome B6F, plastocyanin, which are all like kind of electron transferring proteins and complexes. And once it reaches photosystem 1, electrons are excited again by light and eventually transferred to NADPH. And in the process of all this, it creates a hydrogen ion concentration gradient. And if we remember from cellular respiration, we created a hydrogen ion concentration gradient in the electron transport chain in the mitochondria for the purpose of creating ATP, or not creating, but synthesizing ATP. And we used an enzyme known as ATP synthase, which kind of acts like a molecular motor. So when the hydrogen ions go in, it rotates, phosphorylating the ADP and making ATP. And so we'll take a closer look at how this occurs. So I'm gonna erase this. So right here, this diagram is just the one that was in lecture. And we can see that starting off at photosystem 2, the electrons, you can trace where they go, and eventually they'll end up at NADPH. And in the process, the hydrogen ion concentration gradient is created in the thylakoid space. And when these hydrogen ions pass through ATP synthase, it creates our ATP right here. So these two are used in the Calvin cycle. So if we look at the differences between the ATP synthase and how the hydrogen ion concentration gradient flows between the mitochondria and the chloroplast, it's kind of just the opposite of one another. So for the mitochondria, the hydrogen ions go from the matrix into the inner membrane space and then through the ATP synthase. And for the chloroplast, the hydrogen ions are in the stroma, and then they go into the thylakoid lumen and then back out to the stroma through the ATP synthase. So now moving on to the dark reactions, also known as the Calvin cycle or the carbon assimilation reactions. Basically what happens here is after we get those ATP and NADPH from the light reactions, they're going to be used to utilize or to assimilate carbon molecule that eventually becomes more complex sugar like glucose. So, and there are three main parts of the Calvin cycle. The first is carboxylation, the second is reduction, and the third is regeneration. So they're right here, 1, 2, and 3. And so we'll go through each of them, talk about what happens. So for carboxylation, this is where the CO2 from the atmosphere enters into our into the chloroplast, and this all occurs right here in the stroma of the chloroplast. So the carbon dioxide will enter, and what happens here is there's an enzyme known as rubisco, which is short for ribulose bisphosphate carboxylate oxygenase. So it's pretty long word there, but basically what this enzyme does is it's responsible for the carboxylation of our CO2 molecule into a 5-carbon molecule known as 1,5-bisphosphate. Bisphosphate. Or actually, sorry, it's known as ribulose 1,5-bisphosphate. So this enzyme rubisco will, once the carbon dioxide and ribulose 1,5-bisphosphate get into the active site of rubisco, it forms this 5-carbon or a 6-carbon compound, which eventually will break down into a molecule known as 3-phosphoglycerate or 3-PGA. And since you're breaking down that 6-carbon molecule, you'll get 2 of these. All right, so, and basically that's what happens in our carboxylation part of the Calvin cycle. So moving on to our second part, which is reduction. What happens here is that NADPH acts as a reducing agent, which transfers the electrons from the NADPH to the 3-phosphoglycerate molecule here after it is phosphorylated by this ATP right here. So when we go, so this is where our ATP and NADPH from the light reactions are used. So when we go from ATP to ADP, we lose a phosphate. So it goes from adenosine triphosphate to this adenosine diphosphate. So that phosphate that we lose is going to be in our 3-PGA molecule now. And after that, what happens is the NADPH will transfer its electrons to the 3-PGA molecule, releasing the phosphate and making eventually what we know, what is known as a triose phosphate. Okay, so, and eventually that triose phosphate will go on to make glucose. And this process is known as gluconeogenesis, but I don't think you need to know that. And let's see here. So one important thing to note about this is since we get 2 molecules, or 2 times the 3-phosphoglycerate molecule, we can think of this as for every carbon dioxide that enters the Calvin cycle, we need 2 ATP and 2 NADPH to reduce our 3-phosphoglycerate molecule. So for 1 phosphoglycerate, so for 1 3-PGA, we need 1 ATP and 1 NADPH. And so since we formed 2, basically multiply this by 2, and so we overall for 1 carbon dioxide molecule, we would need 2 ATP and 1 NADPH to reduce the 3-PGA. And another thing to note about this is how I mentioned before that the end product for the Calvin cycle is a triose phosphate molecule. And basically, for every 6 triose phosphate molecules that are produced in the Calvin cycle, we can only utilize 1 out of the 6. And so that leaves us with 5 remaining. Okay, so what's the reason for that? Well, basically, that's what we're going to talk about in our third step. So the last step of the Calvin cycle is known as a regeneration of the molecule that we talked about that combines with the carbon dioxide, known as ribulose 1,5-bisphosphate, or RUBP for short. And for the Calvin cycle to keep on going, we need to regenerate that 5-carbon molecule. So when we produce 6 triose phosphates, 5 of them have to be going back into the Calvin cycle so that we can regenerate it. And this regeneration uses ATP again, and it goes back and forms that 5-carbon molecule. And so overall, for the Calvin cycle, we use 3 ATP and 2 NADPH. So we have, if we break this down, 1 ATP for every 3-PGA molecule. And since we have 2 of them, we'll get 2 ATP and then 1 ATP for regeneration. And 2 NADPH we produce because we need 1 NADPH to reduce, or for every 3-PGA molecule. Alright, so lastly I want to talk about the relationship between photosynthesis and cellular respiration. And so let's start off by writing down the formulas for both of these processes. So for photosynthesis, the chemical formula, we're going to have 6 carbon dioxide, 6 water, which yields glucose. And 6 oxygen molecules. And then for cellular respiration, actually, just right here, it's like the opposite So we're going to have glucose. And we're going to have oxygen, yielding carbon dioxide, water, and ATP. Alright, so if we look at the reactants for photosynthesis, carbon dioxide and water, we notice that those are actually the products of cellular respiration. And similarly, the products of photosynthesis are going to be the reactants of cellular respiration. So plants are autotrophs, meaning that they make their own food. And when they get the energy from the sunlight, it drives this reaction, making glucose. And then the plant breaks the glucose down to get ATP. So in plants are capable of both cellular respiration and photosynthesis. Now, if we think of photosynthesis and cellular respiration in terms of energetics, we can kind of figure out if energy is required or if energy is released in these reactions. So for photosynthesis, we say that we're going from carbon dioxide and water, and we're building up a larger molecule. So photosynthesis can be classified as an anabolic reaction. And cellular respiration, since it's the opposite, where we're breaking down a larger molecule into a smaller one, it can be classified as a catabolic reaction. So, if we think back to what we learned about anabolic and catabolic reactions, usually, most of the time, generally, anabolic reactions are going to have a positive delta G, and catabolic reactions will have a negative delta G. So when we think of the positive and negative delta G's, we can think back to how that is referring to if energy is released or if energy is required. So when we have a positive delta G, that means that it requires energy. And when we have a negative delta G, that means energy is released. And also we can describe these two in terms of endergonic and exergonic. So endergonic overall reaction will require energy. Exergonic overall reaction will release energy. So in the case of photosynthesis, by looking at the delta G, we can tell that this reaction requires energy, or we can call it endergonic. Now, there are some parts of photosynthesis where it is an exergonic reaction because for the light reactions, we do release ATP and NADPH, but overall, we're going to be using that to make glucose, which requires a lot of energy in the process known as gluconeogenesis. And for for cellular respiration, since energy is released, we say that it is exergonic. Okay. And if we look at a free energy diagram of these two reactions, or if you make one, we can say this one's going to be photosynthesis, and this will be cellular respiration. Okay, so in order for photosynthesis to have an overall positive delta G, that means that the energy in our products will have to be greater than our energy in our reactants, right? So delta G is described as G final minus G initial. So if our G final is a larger number than G initial, that'll make this a positive delta G. So what this will look like in our free energy diagram, just generally, and it's just a rough estimation, it's going to be where our reactants here. So if our y-axis is our free energy, and this is going to be our, so the reaction's going that way. So so if the y-axis is our free energy, for photosynthesis, our reactants will need to have smaller free energy in their products, and eventually they will have a much larger free energy in their, they will have a smaller free energy in their reactants and a higher free energy in their products. So products and reactants. All right, and so for cellular respiration, it's going to be the opposite. So we're going to start off with the reactants having a higher delta G, or higher free energy, and then our products moving to a lower free energy.